the setup required for UV Vis. On the right is a spectrometer, and on the left the computer which will be used to analyse the data. First, fill 2 cuvettes with deionized water. This will be used for a reference. It is important to only touch 2 sides of the cuvette, as the other 2 sides must be clear for the beam to pass through. Some cuvettes will have dimples on. That will be the side you handle. The cuvettes are then placed inside the spectrometer and the lid closed.
Ultraviolet visible uses more energy than absorption, so the electrons of metals and non-metal molecules now have motion. Varying wavelength will now find the energy most absorbed. It will take a while to watch this, so don't get bored. For each wavelength absorbed, its complementary colour or wavelength is reflected. So in opposite colours, if yellow is absorbed, then violet will be ejected. Select the most absorbed wavelength and make some standard solutions. Test each one's absorbance to see the calibration curve illusions. Now test the sample and measure its absorbance. Use the graph and find its concentration in accordance.
What is a spectrophotometer? A spectrophotometer is an instrument that measures the changes in light passing through a transparent solution. What happens when light passes through a solution? All solutions and compounds absorb light to a specific wavelength. For example, hemoglobin absorbs blue and green light, so appears red. But why does this happen? Looking at the electromagnetic spectrum, visible light ranges from violet at a wavelength of 400 nanometers to red at a wavelength of 700 nanometers. Colours that we see are the colours reflected by objects, not those that are absorbed. Therefore, leaves on trees appear green because they reflect green light, and hemoglobin appears red because it's reflecting red light. But the spec measures light that is absorbed, or the colours we don't see. So to do an assay on hemoglobin, we would need to set our spec around 500 to 600 nanometers in wavelength. Most specs use light sources within the visible spectrum, but UV and IR specs are also available. A spectrophotometer works by measuring and comparing the intensity of light entering and exiting a sample. Measurements can be expressed as the percentage transmittance, which is the percentage light exiting compared to entering. If all the light passes through a solution, then T is 100%. This would happen for distilled water, for example, where the majority of light would pass straight through. Measurements can also be expressed as the absorbance, A. That is the amount of light taken in or absorbed by the solution. If all the light is transmitted, T is 100%, then A is 0. If no light is transmitted, we would say absorption is infinite. All compounds and solutions have characteristic absorbances. If you were looking at a solution for the first time, you would need to find out its optimum wavelength of maximum absorption, simply by measuring absorption of it at a range of wavelengths. By plotting a graph, the optimum absorption of a compound becomes apparent. So to get the best results from a spec assay, the compound would be assayed at this wavelength.
Ultraviolet visible spectrometry tells us about electronic transitions in atoms and molecules. Spectra are produced when electrons in molecules or atoms move from one electronic energy level to another of higher energy. In doing so, they absorb energy equal to the gap between the two levels. Compounds that absorb in the visible region, such as some transition metal compounds and organic dyes, are coloured. Those that absorb only in the ultraviolet region are colourless. Inside a UV visible spectrometer, there are usually two light sources, one giving out visible light and one ultraviolet. This one is a tungsten lamp, like a car headlamp bulb, for the visible region, and this deuterium lamp gives out ultraviolet. This mirror directs light from the appropriate source into the monochromator. This contains a diffraction grating that acts rather like the playing surface of a CD to split the light into its constituent wavelengths. Different wavelengths correspond to different colours. Red is about 700 nanometers and blue around 400 nanometers. Wavelengths shorter than about 350 nanometers are called ultraviolet. Shorter wavelength light has higher energy. The source produces white light that includes all wavelengths, all colours. The instrument scans through the spectrum, sending different wavelengths of light through the sample in sequence. This is done by the grating, which rotates. A single wavelength passes into the modulator, which consists of a rotor with mirrors on it. This chops the light into two beams. One beam passes through the sample cell, while the other passes through the reference cell. So the instrument is referred to as a double beam instrument. Both sample and reference beams are directed by mirrors onto a detector. This compares their intensities and sends a signal proportional to the ratio of their intensities to the computer that controls the instrument. The logarithm of this ratio gives a quantity called absorbance, which is a measure of how much light has been absorbed by the sample at that particular wavelength. Ultraviolet visible spectra are usually run on solutions. Light does not normally pass through solid samples. Here, we will run the spectrum of a green food dye. To run the spectrum, we place some of the solvent in a sample cuvette to act as a blank, a reference. The cuvettes may be made of glass or plastic if only the visible region of the spectrum is required. Quartz cuvettes are needed for work in the ultraviolet range because glass and plastics absorb UV light. We then place a solution of the sample in a second cuvette. The blank and the sample are placed in the sample holders. The lid is closed to prevent light from the laboratory interfering with the spectrum. The operator types details, such as the wavelength range required and scanning speed, into the computer that controls the instrument. He also zeros the instrument at a point where the sample does not absorb and then starts the scan. The spectrum appears on screen. The data is saved on the computer and a hard copy can be printed. The horizontal axis is normally wavelength and the vertical one absorbance, which is a measure of the amount of light absorbed by the sample. The peaks on the spectrum are the wavelengths of light that are absorbed by the sample, and the troughs are where light passes through. So the dye absorbs orange and blue light and lets through green, so it appears green in colour.